and Ross is really one of the superstars of our movement, really. He's been doing this for a long time, has many interests. He was on with us about a month ago. We talked about longevity and rapamycin, and he really piqued my interest. I actually have got some rapamycin, and I've been peppering him with questions about, what's the proper protocols, what are the combinations, those kinds of things, and it's really a fascinating subject, and it's part of kind of what we do. And tonight, I think for us is going to talk to us about the microbiome, but he's got all sorts of unique perspectives on it. So I'm going to let him introduce himself again, and we really appreciate you being here. And take it away.
Sure. Well, thanks everybody for your interest. Just a little about my background. My original career was pharmacy, and that turns out to be one of the most repetitive, boring careers I can think of. Typing labels and counting pills and slapping labels on bottles hundreds of times a day. But along the way, I got interested in health, and I've been passionate about the science and technology of anti-aging and life extension for probably over 40 years now. For the last 12 years, primary professional focus for me has been on the human gut microbiome. So that's what I'm going to focus on tonight. And let me just share my screen here. And let's see here. Somebody give me the authority to share the screen here. Let's see, screens. And I'm going to get down.
Good habit.
Share. And back here. I think we're making it. Okay, so for those of you that weren't on last time I was presenting here, I talked about rapamycin. There's an image of my book on the left-hand part of the screen. I'm just showing images of two other books that I wrote. Back in 1999, I wrote the Drug-Induced Nutrient Depletion Handbook. Second edition came out in 2001, and I spent 20 years doing presentations to pharmacy groups and medical groups and health organizations, talking about the drugs that are depleted by the nutrients that are depleted by the drugs that people take. And so that's the topic of that book. And when I wrote the Drug-Induced Nutrient Depletion Handbook, I was astounded to learn that oral contraceptives deplete more nutrients than any other class of drugs. So that motivated me to write The Pill Problem, which teaches women about the side effects from oral contraceptives and helps them learn how to prevent them or overcome them. For those of you that are interested in drug-induced nutrient depletions, you can go online and get a free copy of my quick reference guide for drug-induced nutrient depletions by just going to my website, naturalpharmacist.net/DIND, which is drug-induced nutrient depletions. So topics I want to cover this evening: the human gut microbiome ecosystem. I'm going to talk about a two-page paper I wrote titled The Oral Probiotic Dilemma, which really provides the scientific explanation for why orally ingested probiotics don't work. Talk about the importance of broad-spectrum postbiotic metabolites. I'll introduce that term and explain it. And I wrote a paper titled The Microbiome Theory of Aging, and I think Bill has a link to that he can supply everybody else with, or everybody online with. And I really want to spend some time talking about the importance of diet and diversity, the diversity of diet in terms of creating a healthy gut microbiome and this process of fermentation to produce postbiotic metabolites. So this little cartoon is kind of cute. Life is the only game in which the object of the game is to learn the rules. If we apply this to health, it takes on a pretty significant meaning, because there's some pretty basic fundamental rules of health that people have to learn in order to lead a healthy lifestyle. And I know I'm preaching to the choir here, but most of us, myself included, did not have good teachers and good role models when it comes to health. But the human gut microbiome is certainly one of the most critical health regulators that there is in the human body, and people must learn about it in order to lead a healthy lifestyle. And as I'll present information this evening, I'll show you that most people are failing miserably at this. So I start with a deeper dive into the gut microbiome. It's important to understand that over 99.9% of the bacteria that reside in your gastrointestinal tract reside in the colon. So that is really the site of action when we're talking about the gut microbiome. There's also an oral microbiome and an ocular microbiome and a vaginal microbiome, but the real focus for this evening is on the colon and the gastrointestinal microbiome. It's also important to realize that over 70% of the body's immune cells are located in the gastrointestinal tract. So if people have bacterial imbalance and gut inflammation, one of the first things that suffers is your immune system. So I'm sure you all are familiar with dysbiosis. I've just made a slide out of this because a lot of presentations I do in health food stores, a lot of the employees don't understand dysbiosis. The cause of dysbiosis, which is a term, umbrella term for bacterial imbalance in the gastrointestinal system. So bad bacteria are the cause. And the reason bad bacteria are bad is because their metabolic processes cause them to secrete substances that are highly inflammatory. And so here's the symptoms of dysbiosis. You're all familiar with those. A healthy gut microbiome, about 85 to 90% good bacteria, and only 10 to 15% potentially bad bacteria. Everybody has some bacteria that could potentially be bad, but when they only exist in small numbers and the good bacteria vastly outnumber the bad bacteria aren't causing problems. It's when you get bacterial imbalance, and there's so many things in the environment that cause this, toxins in the environment and overuse of antibiotics and so forth, and bad diet. I'll go into all those a little bit deeper, but that's what creates this bacterial imbalance, which causes a lot of health problems. So I'm kind of on the leading edge of unraveling the mysteries of the gut microbiome. My paper that I wrote and was published in June of 2019 is titled Postbiotic Metabolites, the New Frontier in Microbiome Science. That's the first time a paper ever appeared in the scientific literature with the term postbiotic metabolites in the title. So I'm trying to educate the world about how this gut microbiome and these postbiotic metabolites really work in our system. Second paper I wrote, published in Integrative Medicine, January 23, the microbiome theory of aging. And that goes into more detail about all the steps that happen when you have bacterial imbalance. And I'll talk about those steps in more detail as we go along. But we're understanding is that the job of your probiotic bacteria is to convert components in your food into a diversity of postbiotic metabolites. Now, these postbiotic metabolites are compounds that influence every single organ system. And so it's important to understand that it's not probiotic bacteria that regulate your health. It's the compounds the bacteria create, these postbiotic metabolites. Probiotic bacteria are certainly important, but they have to be able to produce these postbiotic metabolites in order to really regulate your health the way they're supposed to. The latest statistics I could get from 2022, Americans spent about $17.4 billion on oral probiotic products. I'm going to try to convince you that most of that is wasted money. And here's why. So I wrote a little 2-page paper titled The Oral Probiotic Dilemma. It's the scientific explanation for why most oral probiotics don't work. There's 3 parts to the oral probiotic dilemma, and the first part is stomach acid. It's designed to kill. It's the front line of your immune system. Stomach acid is anywhere from 10,000 to 100,000 times stronger acid than the acidity in the small intestine and colon. So most oral probiotics are going to hit that stomach acid and be decimated. It's true that some probiotics are enteric coated, and there are some spore-based probiotics that can survive gastric acidity. This still does not ensure that they're going to be effective, and I'll talk about that in my next bullet points here. So dilemma number 2 is the small intestine, also a very hostile environment for bacterial survival. We secrete bile acids and pancreatic digestive enzymes into the small intestine. Their job is to digest food. They'll destroy the cell wall and cell membrane of bacteria if they make it that far. And the third dilemma is, I think, one of the most serious sins against mankind or humankind in the history of the world. People eat crappy diets. And so it's important to understand that dietary fibers and polyphenols are the two primary food groups for your probiotic bacteria. And they occur primarily in plant-based foods. Fruits and vegetables are the primary sources. There's some polyphenol. In fact, there's over 8,000 polyphenols that have been structurally identified. But in addition to fruits and vegetables, there's some polyphenols in herbs and spices and some in whole grains. But the primary sources of these foods for your probiotic bacteria are fruits and vegetables. So the next two bullet points are a compilation of multiple studies that have been published. 90 to 95% of American children and adults do not meet the RDA daily recommended allowance for fiber intake. Different studies report from 80 to 91% of Americans fall short in every color of phytonutrients consumption. So these are the polyphenols. So in these two bullet points, I hope you grasp that almost everybody in the United States is not consuming a diet that supports a healthy gut microbiome. You've got anywhere from 800 to 1,000 species of bacteria in your gastrointestinal system, primarily in your colon. I like to do an analogy between your gut microbiome and the Amazon rainforest, two of the most complex, diverse ecosystems on the planet. In the Amazon, you've got thousands of different species of insects and mammals and birds and reptiles. All those animals require different types of food. The different species of bacteria in your gut microbiome have different DNA. They're like different animals. They require different types of food, different types of fibers, different types of polyphenols. So it's critical that people ingest a diversity of plant-based foods, and almost nobody is doing that. So it's not just the quantity of dietary fibers and polyphenols that people aren't ingesting, but it's also the diversity. I try to get people to understand the importance of eating a little bit of as many different types of fruits and vegetables every day as you can. Every time you eat, you're hosting a very large party. You're feeding 100 trillion guests. So here's the standard American diet. This burger on the left, people are getting a little lettuce and a little tomato, maybe a little slice of onion. That's not a very wide diversity of polyphenol and dietary fiber containing foods. The image on the right, commercial grocery stores, all of those packaged and processed foods. Americans are seriously lacking in the diversity of intake of these dietary fibers and polyphenols. Research published in the journal Nature Food reports 73% of the US food supply is ultra-processed food. And on average, ultra-processed foods are 52% less expensive than healthy alternatives. So that's the story. That's what's really one of the primary reasons why so many people have gut problems, and it morphs into a wide range of systemic health problems. Here's one of my analogies between probiotics and the gut microbiome. I'm comparing it to a manufacturing plant in an automobile processing plant. So in your automobile manufacturing plant, you've got hundreds of workers, but they require thousands of parts to build vehicles. The analogy in your gut microbiome, your probiotic bacteria are the workers, but they have to have a diversity of the dietary fibers and polyphenols to produce the postbiotic metabolites. So emphasizing the importance of balance and diversity and using these analogies again, in the automobile manufacturing plant, if you've got thousands of new workers coming into the plant, but they only have one skill, like they can just put on windshield wipers, that really messes up the balance and the diversity of the whole manufacturing plant. Similarly, in the Amazon rainforest, if all of a sudden you put millions of new monkeys in the Amazon, that's going to really mess up the balance and the diversity of the ecosystem. And it's the same in your human gut microbiome. People taking these high-dose probiotics are working against balance and diversity. And Americans tend to think more is better. So there's competition. Mine's got 50 billion, mine's got 100 billion, mine's got 200 billion. These products are not providing the benefit that manufacturers lead them to believe. In many cases, they're really working against balance and diversity. There's one study, I don't have my slide in here this tonight, but there's a study that was published saying high-dose probiotics can actually cause your immune system to mount an alarm reaction. It's not that they're bad bacteria, it's just too much of one or several strains that throws things out of balance. Another analogy here, temperature versus acidity. We all know that temperature is a critical regulator of the environment for the health of plants and animals and humans. The analogy in the gut microbiome, it's acidity that is the critical regulator of the microbiome ecosystem. And the optimal acidity is from 6.2 to 6.8. You're all familiar with the pH scale, 0 to 14, 7 is neutral, so 6.2 to 6.8 is weakly acidic. So my question is, how do people achieve and how do people maintain this weakly acidic pH in the gastrointestinal ecosystem, which is the optimal pH to promote the growth and proliferation of probiotics, and it suppresses the growth of pathogens? Well, it turns out a lot of these postbiotic metabolites are weakly acidic compounds. Your short-chain fatty acids, propionic, acetic, butyric acids, organic acids, nucleic acids, amino acids, folic acids. If you eat a diverse diet, your bacteria will produce these weakly acidic compounds and you'll have a healthy microbiome ecosystem. But again, I emphasize, most people are not eating the diet that allows this to happen. Let me just summarize some of the things I've already said. Thousands of animals in the Amazon, thousands of species of bacteria in your gut microbiome. Consuming high-dose probiotics doesn't fix a damaged gut microbiome ecosystem. And it's important to realize the foundation of a healthy ecosystem, whether it's the Amazon rainforest, the coral reefs in the ocean, the gut microbiome in your colon, food is the basis of a healthy ecosystem. Food allows the animals or the bacteria to thrive and survive and develop. And then in the gut microbiome, the diversity of bacteria can create the diversity of postbiotic metabolites that are the master regulators of your health. Now I'm going to shift gears and talk about a manufacturing process that was created in Japan using natural foods and pure spring water. And it's a multi-year fermentation system to produce a broad spectrum of postbiotic metabolites. So in this system, those are 80-gallon fermentation vats. Sterile manufacturing facility in southern Japan. They start out with 12 strains of probiotic bacteria that are the starter strains, a multitude of Japanese fruits and vegetables and mushrooms and seaweeds, a diversity of dietary fibers and plant polyphenols, 3-year natural fermentation process. And at the end of the fermentation process, the product has got over 500 of these postbiotic metabolites. There is no other product in the world like this. This is a slide showing the diversity of Japanese fruits and vegetables and mushrooms and seaweeds that are used in that manufacturing process. And the right-hand slide image on the slide there is I'm just listing the different strains of starter bacteria that are used in the production process. And so in this multi-fermentation year fermentation process, first on the image on the right-hand slide, I've got fermented foods. We all know that fermented foods are healthy types of foods. And we all know that they have kind of a tangy, tart taste. That is due to the postbiotic metabolites that are created during the fermentation process. So I want to emphasize that the production process in the image of the center of this slide mimics the natural fermentation process that goes on in the colon. So this Japanese production process that I'm talking about really mimics nature. It takes food anywhere from 24 to 72 hours to pass through the colon before it gets eliminated in the bowel movement. So that's the amount of time your bacteria in your colon have to break down the food and produce these postbiotic metabolites. In this multi-year fermentation process in the center image, 3 years is 26,280 hours. And the bacteria are provided with a multitude of fruits and vegetables and mushrooms and seaweeds, a diversity of dietary fibers and polyphenols. Over here in the human gut microbiome, almost nobody is consuming that diversity of plant-based compounds that allows a diversity of postbiotic metabolites to be produced. And because of that, we have an enormous number of health problems. I'm fond of saying we have an epidemic of epidemics. Heart disease, cancer, diabetes, obesity, autism, Alzheimer's disease, inflammatory bowel disease. None of these were epidemics when I was in high school back in the 1950s. I think the health of humanity is going down seriously. And I think that this gut microbiome problem is one of the main contributors to bad health. Those in the center image are the 80-gallon fermentation vats that are used in 3 years of the manufacturing process. There's a second product that undergoes an additional 2 years of anaerobic fermentation in these mammoth 10,000-gallon anaerobic fermentation vats. Remember, 99.9% of your bacteria reside in your colon, which is an anaerobic environment. So this 2 additional years of anaerobic fermentation gives the bacteria that are capable of producing postbiotic metabolites in an anaerobic environment 2 additional years to produce those metabolites. So I emphasize there's 2 pieces to the puzzle. Probiotic bacteria are extremely important, but you have to feed them well or they won't thrive and survive. This is a slide just showing my 8-minute YouTube video. You can Google Ross salad buzz and watch it. But what I'm teaching people is how to make a salad, which is a meal that really supports your gut microbiome. My lettuce mix that I buy from my health food grocery store locally here in Ashland, Oregon, has got 4 different types of lettuce in it. And then you can see I've got added vegetables. So I got 16 different types of fiber and polyphenol containing foods in my salad buzz.
It's on our email from this week, Ross, announcing that you're going to be on. I have the video on there. All I have to do is click on it.
Click on the video.
It's on the email that we sent out for this week's announcement that you're going to be on. I have your salad buzz.
Okay, so people will have access to it. Good, thank you. So the vegetables I got there are not set in stone. Different times of the year, there's some different foods available at seasonally different times. So it's just teaching people the concept of how to get a diverse range of these plant-based foods into your diet every single day. The secret to this process is that when I finish processing my salad buzz once a week, I squeeze a lemon on it and toss it with the lemon juice. Lemon juice has vitamin C, antioxidant, so my salad buzz will stay fresh in a Tupperware container in the refrigerator for up to a week. So this is a tremendous time saver for people, because when I make a salad most evenings for my wife and myself, I got 2 bowls on the counter. I go to the refrigerator, I pull out my lettuce bag, I pull out the Tupperware with the salad buzz. It's a handful of lettuce, a handful of salad buzz, maybe some wild-caught salmon or some garbanzo beans for a source of protein, but it takes like 2 minutes to make my salad for dinner. And you couldn't do anything better to support your gut microbiome. So these are the different categories of postbiotic metabolites. Different strains of bacteria are capable of producing all of the B vitamins, vitamin K and folic acid, some of the essential amino acids. You know, we used to think that we got all of our nutrition from the food we eat. Now we find out that our bacteria are capable of producing a lot of these nutrients for us if you're eating a healthy diet. I talked about the short chain fatty acids. They make a number of immune system regulating compounds. This is an important point. These natural antibiotics, they're called antimicrobial peptides. 2 different classes of them, bacteriocins and defensins. These are short chain amino acids. They're small proteins. that different strains of bacteria will produce, and they kill pathogens, but they don't harm any of your good bacteria. It's one of the most important parts of your immune system, these natural antibiotics that your probiotic bacteria will make. Neurotransmitters, everybody knows about a deficiency of serotonin and increasing the risk for depression, and the pharmaceutical industry did something that drives me crazy, creating a multi-billion-dollar a year category out of the select serotonin reuptake inhibitors. 95% of the serotonin made in your system is made in your gut by your bacteria. And then there's gut-brain communication, or that gets some of it transferred to your brain. Antioxidants, cell signaling compounds. So we're just starting to learn more and more all the time about these postbiotic metabolites. So this is just some of the primary categories of metabolites that are known to date. And now I'm going to talk about some of the activity of these various postbiotic metabolites, showing a highly inflamed colon up here on top and a healthy one down in the bottom. So the person up on top here, they would like to have some anti-inflammatory activity. And a lot of the postbiotic metabolites do have anti-inflammatory activity. I mentioned the antimicrobial peptides that will directly kill pathogens. The reason that person has so much inflammation is they have bacterial imbalance, and too many bad bacteria are producing inflammatory compounds. This person with this inflammatory situation in their colon would also like to accelerate the growth of new epithelial cells. On average, healthy people create an entirely new lining of their gastrointestinal tract every 6 to 10 days. Scientists estimate that about 10% of our total daily energy expenditure as humans is the energy that goes to producing and renewing epithelial cells in the gastrointestinal tract. Butyrate, one of the primary short-chain fatty acid postbiotic metabolites, is the primary energy source for renewal of epithelial cells. I talked about the acid-base balance. You're going to accelerate healing of the gut microbiome lining. There's this intestinal barrier, and when that is inflamed, you get leaky gut, and I'll talk about that in a little bit. The immune system is regulated by this, detoxifying toxins, improving GI symptoms. Nothing will improve GI symptoms faster than directly ingesting these postbiotic metabolites. We know about essential nutrients, amino acids, fatty acids, vitamins, and minerals. These are compounds we can't make. We have to get them from our diet. I'm proposing that we need to rethink this concept of essential nutrients. These bacterially derived postbiotic metabolites need to be recognized as essential nutrients. We don't make them, but our bacteria make them. So fatty acids, amino acids, vitamins, and minerals come from the diet. The postbiotic metabolites come from the diet, but they're produced by your probiotic bacteria. I want to say a couple words about antibiotic resistance. It's a huge global problem and getting worse. Bacteria are among the fastest responding organisms in the world. They double about every 20 minutes. So 3 times per hour, a bacteria can double. We think about humans, one generation being 20 or 30 years, maybe 1 to 4 children. Bacteria double 3 times per hour. In 24 hours, 72 doublings. A 7-day course of antibiotics, 504 doublings. So you can see these bacteria reproduce and multiply so fast, they can develop resistance to antibiotics very quickly. World Health Organization has ranked antibiotic resistance as a global crisis. Let me get rid of that, excuse me. So I'm going to show a short little video that I hope is motivation to be proactive. I usually do this to non-professional audiences, but I thought you might find it interesting also. Just it emphasizes how important it is to be proactive to create and maintain a healthy gut microbiome ecosystem. So this short little 2-minute video was done at Harvard. And if you want to search it and watch this again, I can send the link to Bill. You search mega plate. So on the right here, I'm showing a 3-inch diameter Petri dish with agar, which is the growth medium that they use for bacterial growth. So why am I showing this video? Emphasizing that dysbiosis causes a wide range of gut problems, which are often treated with antibiotics. And dysbiosis also causes a weaker immune system, which is going to cause more systemic wide health problems that often get treated with antibiotics. And I'll spend a little bit more time on this in some of the future slides. Dysbiosis causes gut inflammation, which damages the intestinal lining, which allows lipopolysaccharides to leak into our system. And that is a huge health problem. And I'll talk about that in a minute. So here's the little video. If I can get it to play here.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, porous and thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. Then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So I think you'll agree that really emphasizes how incredibly important it is to create and maintain a healthy gut microbiome. I'm sure many of you have heard of Alessio Fasano. I hope he wins the Nobel Prize sometime. He actually discovered zonulin, and zonulin is a compound that is produced when you have gut inflammation. And tight junctions between your epithelial cells, the one cell layer thick of cells in the GI tract, are held together by proteins on the cell surface that kind of act like Velcro. So these proteins on the cell surface and your GI tract are what hold your epithelial cells together and create tight junctions. Substances are supposed to be able to traverse through cells from your GI tract into systemic circulation, but things are not supposed to go in between the cells. But when you have gut inflammation, zonulin is produced. Zonulin destroys the proteins that are your tight junctions, degrading the intestinal tight junctions and allowing lipopolysaccharide to leak from your GI tract into systemic circulation. Now, lipopolysaccharides are some of the most highly inflammatory substances known to man. And in the next 3 slides, I'm going to go into detail about a really phenomenal study that was conducted. And there's the title of the study, Human Model of Inflammatory Cardiometabolic Dysfunction, Double-Blind Placebo Crossover Trial. So this is a study in which people were injected with low-dose lipopolysaccharides. one of the important parts of the study design, it was a crossover trial. So in the first part, part A, group 1 got injected with low dose lipopolysaccharides, second group's a placebo. These people are in a medical facility for 36 hours with tremendous amount of documentation and observation, all sorts of blood work and all sorts of questionnaires, how are you feeling, any symptoms and so forth. Then there's a washout period of several weeks, and then they reverse. And group 2 now gets the low dose injection of lipopolysaccharide, and the people that got IV lipopolysaccharides the first time is now the placebo group. So here's the outcome. 10 healthy volunteers, 36-hour inpatient visits, blood work, questionnaire. The thing to emphasize here, the dose of lipopolysaccharides was so low, nobody had any symptoms. But when they did the blood work, low-dose polysaccharides resulted in a 25-fold increase in tumor necrosis factor alpha, a 100-fold increase in interleukin-6. These are inflammatory markers, which are telling us that when you have just a little bit of leaky gut and a little bit of lipopolysaccharide is leaking into your system, so little that people don't have any symptoms, they still have systemic endogenous inflammation that's accelerating biological aging. So it is so critical to get people to understand the importance of this diversity of plant-based foods to support a diverse range of probiotic bacteria, so you get this diversity of postbiotic metabolites that regulate your health. So in this slide, I'm asking the question, what happens when you damage the ecosystem? In the Amazon rainforest here, if you put the ants and the birds, and the bees and the butterflies and the monkeys and the snakes back into that damaged Amazon ecosystem, those inhabitants are not going to thrive and survive, and they're not going to cause the trees to regrow. My point here is you can't just put the inhabitants back into a damaged ecosystem and expect to fix the ecosystem. So in the inflamed gut microbiome, where you have dysbiosis and bacterial imbalance, just taking oral probiotics doesn't fix the problem. Here, I'm just making the point on the left that if orally ingested probiotic bacteria do survive transit through the gastric acidity in the small intestine, when they reach the colon, they have to be able to find dietary fibers and polyphenols and start the process of converting them into postbiotic metabolites. That all takes time. And I presented slides earlier with summarizing studies that say 80 to 95% of people aren't ingesting the types of foods that allow their bacteria to make the postbiotic metabolite. Over on the right-hand side of the slide, I'm just showing that when people take this product with a high diversity of postbiotic metabolites, they immediately start to get anti-inflammatory activity. The antimicrobial peptides are killing pathogens. They rebalance the acid-base level, accelerate the regrowth of healthy cells and the lining the GI tract. All that starts to happen pretty quickly. Emran Mayer, very famous MD, gastroenterologist and microbiome scientist. This is his second book. I made a slide out of a comment that he made in his book. Taking the popular and highly advertised shortcut of popping a daily supplement pill containing billions of colony-forming units will not do the job. So it's not just me that's promoting this manufacturing process that has a diversity of postbiotic metabolite. Here's Emran Mayer saying these high-dose probiotics don't work. I'm going to just quickly review several studies that have been done on these products where there's a high diversity of postbiotic metabolites. This is the vitamin C knockout study with mice. Normal mice live about 24 months. Scientists created a strain of mice where they knocked out the gene responsible for producing vitamin C. So the vitamin C knockout mice can't produce vitamin C, accelerated free radical damage. They only live on average about 6 months. So scientists did a study comparing normal mice with the vitamin C knockout mice, and then they gave the vitamin C knockout mice this high potency, diverse postbiotic preparation. And instead of getting a deranged microbiome and decline in postbiotic metabolites and a significant decline in ATP energy production, when they can't produce vitamin C, but they're getting the postbiotic metabolites, same microbiome, normal production of postbiotic metabolites, actually higher production of ATP than the normal mice. So pretty powerful stuff when you get these postbiotic metabolites. Here's a study on constipation, widespread problem in America. Approximately 20% of adults, 33% of elderly people over 65 have chronic constipation. So this is a study done with female college students who have a long history of constipation, and a control group, which are age-matched women with no history of constipation, college-age women. And so they took 3 capsules daily of either the placebo or the product with the 500 plus postbiotic metabolite for 2 weeks. At the end of 2 weeks, all the women who had a long history of constipation had normal bowel movements. So these postbiotic metabolites aren't used just to treat diarrhea. They can also be used to treat constipation. These products are adaptogens. They normalize bowel function. There's the reference for that. This study was done on 6 long distance runners on a track team in Australia. The men did an intense run. Then they took 3 capsules per day of the postbiotic metabolites and then did a follow-up intense run. 8.4% increase in hemoglobin. Now, if you know anything about professional sports, doping is illegal. It's illegal to take drugs that increase your hemoglobin level. Here, we're showing that if you take these postbiotic metabolites and normalize gut function, you'll increase hemoglobin levels. Increase in VO2 max, so it increases lung function, increasing the lactate threshold so they can exercise intensely for longer periods of time before lactate builds up in the muscles, increasing their cardiac output significantly. When you increase your gut microbiome ecosystem, everything works better. This is bone density study in Japan, 2 years, ages 20 to 60. The dosage was interesting, from 3 to 10 capsules a day for 24 months. The dosage difference is due to the fact that some Japanese individuals are very small, petite people. Some are big, mammoth, sumo type wrestlers. So the dosage was based on their size and their weight. But look at the results here. In 2 years, all the men and the women had significant increases in bone mineral content and bone mineral density. This was an observational study. So the scientists conducting the study didn't give any mechanisms of action. But my theory is that we know that minerals like calcium and magnesium have to have a slightly acidic condition for absorption. And all those postbiotic metabolites, our weakly acidic compounds, are going to promote the absorption of these minerals that are involved in bone metabolism. And the B vitamins are involved in bone metabolism. Many of your probiotic bacteria are producing those compounds. So that's my theory on this outcome. Here's a study that was published in the Journal of Functional Foods back in 2022. It was a test of fatigue and with this high-dose probiotic product, and they measured fatigue on 2 different rating scales. And I want to emphasize that only totally healthy people could be participants. No high blood pressure, no obesity, no history of cardiovascular problems, no depression or anxiety. Totally healthy people were the participants here. 12-week clinical trial. This is the visual analog scale, where once a week for 12 weeks, they just marked an X on the scale, which was their subjective feeling of how fatigued they felt during that week. And the other rating scale is the chronic fatigue score, 11 questions every single week for 12 weeks. And you see the outcome here, significant reductions in fatigue in both of these rating scales. And again, this is healthy people. So if people with health problems are doing this and rebalancing their gut microbiome with these postbiotic metabolites, they'll get a much bigger bang for their buck. Advice following antibiotics. I think people should take these postbiotic metabolites during the course of antibiotics, but don't have them in the stomach at the same time. maybe take them a couple of hours after a dose of antibiotics. And then following the course of antibiotics, I strongly recommend that people double or triple the dose of these postbiotic metabolites for 10 days to 2 weeks to accelerate the rebuilding of a healthy gut microbiome. And what age can people take ingesting these postbiotic metabolites? As soon as a child can comfortably swallow one of the capsules. And one thing I emphasize a lot for women that give birth to an infant via C-section, bite this capsule, squeeze the paste out, rub it around in the inside of your infant's mouth. You want to help that infant build a healthy gut microbiome. And if he or she was born in a cesarean section birth, they did not get exposed to the mother's vaginal microbiome. A number of dentists are starting to recommend that people take one of these capsules with postbiotic metabolites and bite it and squeeze the contents out and swish it around in your mouth for about 20 seconds or so before swallowing it. A lot of dentists are recommending that you do it at bedtime. It supports a healthy oral gut microbiome. And the image on the right here is showing somebody with a nasty abscess in their teeth. One of the people I work with tells a story of a friend of hers who had an abscess like this. It was a holiday weekend. She couldn't get to the dentist. And Kathy told her, bite these capsules, squeeze the paste out, smear it on your tooth, do it every hour. And the abscess totally cleared up and she never had to go to the dentist. These postbiotic metabolites, these weakly acidic compounds suppress the growth of pathogens. The antimicrobial peptides kill pathogens. This stuff really works. I did CrossFit for 5 years. I don't do it anymore. I'm too old. But when I was in CrossFit, one of my buddies had MRSA in the nostril of his nose, and his 4-month-old baby daughter had a big patch of MRSA on the back of her right thigh. He was petrified about taking his daughter to the hospital and having her blasted with antibiotics. I said, Josh, try for a couple of days just squeezing this paste out of this product and smear it on your nose and on your daughter's bad spot on the back of her thigh. It all cleared up in 4 to 5 days. His nose cleared up in a couple of days, and it took a little longer for his daughter's MRSA to clear up. But we can't go publicly and make this claim, but these things really work. These postbiotic metabolites have powerful activities against pathogens. A couple slides in closing here. This is a comment that was made in a study published in 2022. The author is stating there's increasing evidence that the health benefits associated from probiotics are typically derived from the metabolites and secreted compound produced by the live microbes rather than the presence of the live microbes themselves. So it's the postbiotic metabolites, not the bacteria themselves, that are regulating your health. In 2016, Cleveland Clinic did something very interesting. For many years, they've been voted as one of the top hospitals in the United States, and they've been the #1 cardiac surgery unit in the United States for, I think, the past decade. So in 2016, Cleveland Clinic got together a panel of over 100 top scientists and physicians and asked them to research the medical literature and look for medical innovations that are likely to be game changers in the near future. They had a list of over 200 medical innovations. They narrowed it down to their top 10. There was a huge conference with over 500 physicians, scientists, journalists, big coverage of this conference or this presentation. And they started at number 10. And when they got to number 1, the #1 game changer, using the gut microbiome to prevent, diagnose, and treat disease. So this is really a critically important health topic. This is, people can get more bang for their buck eating a healthy diet or switching to a healthy diet and taking these postbiotics to create and maintain a healthy microbiome than many other things they spend time on. And one other study from 2021, the author stated, it's now clear that the depletion of microbial metabolites beneficial to host health is associated with a rapid rise in non-communicable chronic diseases. So again, it's a bad gut and intestinal permeability that's leading to this epidemic of epidemics of health problems. It all goes back to the gut. Hippocrates is credited with the saying, all disease begins in the gut. I don't think anybody really heard him say that, but it's the right idea. So this product and this production process, this fermentation process, produces a broad spectrum of postbiotic metabolites. There are some probiotics in it when it's encapsulated and some prebiotics from the fermentation product, but the vast majority of this fermentation product is the postbiotic metabolites. A little history about how this product was created. There's a man by the name of Ichiro Ohira, a Japanese man. When he was a young man, he was a Buddhist monk, lived in the monastery for years. And when he graduated, he left the monastery, enrolled in college. He got a degree in agriculture. And after he graduated, he developed a business and a career as a landscape architect. He became incredibly successful as a landscape architect. He had a global recognition of his landscape architect career. In 1976, the country of Malaysia invited him to create and design what they called a large freedom park for one of the capital cities in one of the states of Malaysia. Ohira accepted that job, and 4 years later, 1980, he was invited back to the grand opening ceremony. He came back to Malaysia, attended the grand opening ceremonies, and he ate some tainted food and got violently ill. He was writhing on the ground, nausea and vomiting. There happened to be a shaman attending that grand opening ceremony. He sees Ohira lying on the ground in obvious distress, kneels down, cradles Ohira's head in his lap, reaches into his leather satchel, and starts to spoon feed Ohira some black, syrupy, pasty stuff. Ohira is astounded at how quickly he recovers. So he asks some questions, finds out that this product that the shaman had administered to him was created from locally fermented Malaysian fruits and vegetables. So that's where Ohira got first exposed to this concept of fermenting fruits and vegetables. He takes samples of it back to Japan. He plays around it for a couple of years, and he gets totally frustrated because he doesn't have the scientific background to study it the way he'd like to. So at age 49, with a globally successful career as a landscape architect, he totally abandons that career, goes back and enrolls in the PhD microbiology program, gets a PhD in microbiology, and after graduating, it took him 10 years to develop this fermentation system, experimenting with which types of food work best, which strains of bacteria work best, how long does the fermentation have to be? And so that's the history on how this whole fermentation process got developed. There is no other product in the world like it. This is the #1 product in the world for the direct delivery of postbiotic metabolites. So these are some of the key benefits that people get when they start taking this fermentation product that has a broad spectrum of postbiotic metabolites. Everybody's going to get a healthier gut microbiome. I mentioned that the studies on fatigue. I'll mention one other study that was done. We don't go on social media to talk about this study because the animal rights people get all upset with us. But this was a mouse study. It's called the swimming test to fatigue, swimming test to exhaustion. So there's this big swimming tank, and they have mice, divide them into 2 groups. One group gets Dr. Ohira's, the other group gets the placebo. And once a week, for 4 weeks, they put a single mouse in the tank and it swims until exhaustion. They measure the time, they rescue the mice, they're treated humanely, they don't allow them to drown. But after 4 weeks, the mice getting Dr. Ohira's probiotics swim 2 times longer than the placebo controls. If you get your gut microbiome working better and you digest your nutrients better, you get more energy, more stamina, less fatigue. This is one of the things that people really notice when they start taking this fermented product with a broad spectrum postbiotics. And improved intestinal function and better elimination. I mentioned the statistics on how widespread chronic constipation is. Many people that start taking this are really literally aware of having better elimination. More strength, more stamina, more endurance, stronger immune system, better pH established, active against pathogens. I mean, all of this happens with these postbiotic metabolites. So what we try to get people to do is just try a 30 count box of this high dose postbiotic metabolite. And we encourage people to start taking double dose, 2 capsules twice a day for a week. 30 count box, 2 a day, twice a day is 28 capsules. So that'll last for a week. Most people really experience a difference. And for the participants that are on this presentation on this call tonight, those of you that live in the United States, we're going to send you a gift package that has some of these products in it. And we encourage you to try it. Try taking 2 capsules twice a day and let me know how you feel. So that's the end of my presentation. I appreciate your interest and the opportunity to be with you this evening. I'm happy to respond to questions if there are any.
Great as always, Ross. Thank you so much.
Yeah.
There's questions in the chat. Why don't we look at those first?
Yeah, let's see here.
Okay, what's your opinion on fulvic humic acid products?
Oh, yeah. Okay. It's pretty interesting. Every year, the people that make this fermentation product that I talked about tonight do a scientific presentation in a different city every year. In 2017, the scientific meeting was in Vancouver, Canada. And at that meeting, Dr. Muniaki Takahata, who is actually the lead scientist for this company, gave a really wonderful and very interesting presentation. And he said, through an analysis, they discovered that the largest single ingredient in this multi-year fermentation product are fulvic acids. Now, I had never heard of fulvic acids before, so of course I dove into the research and have learned about them. And they are really interesting compounds. There's humic acids and there's fulvic acids. Fulvic acids are much smaller. And the unique thing about fulvic acids is that they have more binding sites per molecule than any other substance in nature. So one molecule of fulvic acid can bind to 12 to 15 minerals. And because they're nano-sized particles, they're easily absorbed across cellular membranes. So they're a major mechanism of transporting minerals into cells. They're primarily produced in nature. Bacteria break down organic matter in the soil and produce fulvic acids, and then the fulvic acids help transport minerals into the root hairs of growing plants. They don't occur naturally in humans because there's not enough time. But in this fermentation process that I showed you today, 3 to 5 years of fermentation, the bacteria have enough time to break down food substances and produce fulvic acids. So I think that may be one of the main mechanisms for why this broad spectrum postbiotic metabolite product from the multi-year fermentation process, I think some of the benefits are due to the fact that it contains these fulvic acids, which will help absorb minerals into your system.
Okay, great.
And one other comment about that. Bacteria break down organic matter in the soil to produce fulvic acids, which are critical regulators of nutrients coming into plants. One of the greatest sins against mankind is the spraying of all these agricultural pesticides, insecticides, and herbicides on agricultural crops, which very little of it goes onto the crop. Most of it goes into the ground and kills the bacteria. So the bacteria can't break down the organic matter and produce the fulvic acids. And so you get weaker plants, and then you have to have more pesticides, and it drives me crazy. Enough said on that rant.
Okay. I'm not sure I should even read the next one. It says, I'm seeing Dr. Clearfield this week, but what do I do about my candida bowel overgrowth, thrush infection in the throat?
One of the strains of bacteria in this multi-year fermentation process. I didn't put in slides on this, but it's called Enterococcus faecalis TH10. This is a strain of bacteria that was discovered by O'Hara in Malaysia. He discovered it in the Malaysian fermented food tempeh. And he studied it for years. And it is one of the most powerful antibiotics that's ever been discovered. And it's just a probiotic. But they've discovered the reason it's such a powerful probiotic bacteria, it produces a postbiotic metabolite called phenylactic acid. And there's studies that have been done with a 1% solution of this phenylactic acid. It kills all sorts of pathogens. It destroys the cell membrane of E. coli and Candida and MRSA and on and on and on. So this is such an important strain of bacteria to that multi-year fermentation process that I showed you. They add a specific amount of that particular strain of bacteria to the final batch before they encapsulate it. So my suggestion about Candida, if you've got Candida in the mouth, I would chew these capsules and swish it around in your mouth and then swallow it. And when it gets into your digestive tract, especially the colon, it's going to fight Candida. I don't have any studies. I can't make the claim that this is a treatment for Candida, but we do have cell culture studies that show that Enterococcus faecalis TH10 and the phenylactic acid do suppress the growth of Candida. So that's one recommendation for Candida.
Any interaction with the, antifungals at all?
Yeah, it's also got some antifungal activity. And here's an important point to make too. I mentioned the story of Dr. O'Hara getting sick and ingesting and getting well very quickly. A lot of people who get food poisoning, you go to Mexico and they ingest bad water or they ate bad food, nausea and vomiting. People would take 10, 12, 15 capsules of this, bite it and squeeze the contents out so the contents get directly into your stomach. Because the capsule itself is a patented capsule made by the Fuji Capsule Company in Japan. It stays hard in the acidic environment in the stomach, and then when it reaches the small intestine, which is a much more alkaline pH environment compared to the gastric acidity, it dissolves and releases the contents in the small intestine. But if you got food poisoning in your gut, you want those contents to get into the stomach. So 10, 12, 15 capsules, chew it, squeeze the contents out, and then swallow it so it gets directly into your gut.
Okay. All right. What is your opinion of oral akkermansia?
It's got a lot of good studies on it. And one thing I would like to do, or like somebody to do, I want to know if the benefits for akkermansia are due to the bacteria itself or due to a postbiotic metabolite it's producing. I haven't seen any research on that, but most of these bacteria, when they're taken orally, are going to get killed and not provide much benefit. But I know there's some good research on akkermansia, but I'd like to know the mechanism of how it's providing the benefits.
Okay. A couple of comments. I'm not going to mention it, but these products are available on some supplement distribution websites. It's in the chat if you want to see it, what it is. In the Gerson protocol for cancer, 5 coffee enemas are recommended. Would that have any effect on the microbiome?
Yeah, that's an interesting question. I actually worked as a consultant for a year with the Gerson organization, and living in San Diego, many trips to their clinic down in Mexico. So I'm very familiar with their protocol. When you do coffee enemas, the caffeine gets absorbed through the rectal hemorrhoidal vein and goes directly to the liver, dilates the rectal hemorrhoidal vein, and you dump toxins from the liver very quickly. So it's a very effective method for quickly detoxifying the liver.
Okay.
But you're also going to flush out your microbiome in your rectum and your colon. So I wouldn't do that long term, but for short term, it's very, very effective. They used to recommend that people do liver juices, but I think they've stopped that now because they can't get healthy liver to process. And they use the Norwalk juicer. And my problem with that is that you're eliminating all the fiber. And so I know that there's some wonderful results with the Gerson organization, and I worked with them for a year, and I've seen wonderful results. But my focus is a lot on the gut microbiome, and you have to get the fiber as in order to have a healthy gut microbiome. But if you're dying of cancer, you might want to detox your liver before you start to focus on your gut microbiome.
I guess these postbiotics seem to be available in the Philippines. Just want to clarify, is 1 week enough for a patient with leaky gut and brain issues?
Yeah, that's right. They are marketed in the Philippines, and nobody really knows the answer to what dose or how long is required. I mean, biochemical individuality, and everybody's got a different diet, and so we really don't know how long it takes to heal a damaged lining in the intestinal barrier, but we understand the mechanism that's required, which is reducing inflammation, getting rid of the bad bacteria which are causing the inflammation, getting more of these postbiotic metabolites, especially butyrate, which is the primary energy source, to renew those epithelial cells. But in terms of what dose and how long, nobody knows. Haven't done those studies.
Is there any effect of these postbiotics on SIBO?
That's an interesting question. SIBO is a difficult situation, different situation. I'm sure most of you know what it is. It's bacteria from the colon have relocated in the distal end of the small intestine. So it's not necessarily a condition that's caused by bad bacteria. It's bacteria that have moved into the neighboring neighborhood or are living in the wrong housing development. They're in the wrong physical location in the gastrointestinal tract. And most people with SIBO cannot tolerate probiotics because the bacteria will ferment the foods in the small intestine and create gas and bloating and pain and diarrhea. There's a couple of different therapeutic diets for SIBO, the FODMAP diet, the specific carbohydrate diet. They should only be used for short periods of time. I would never encourage people to be on those diets for long periods of time. On those diets, many foods that are normally thought to be healthy people are recommended to avoid them. That's because they're going to cause fermentation. With this product that I've talked about tonight, the postbiotic metabolites, most people can take this product if they have SIBO because it's not delivering probiotic bacteria. Very low levels of probiotic bacteria compared to the commercial products that contain tens or hundreds of billions of colony forming units. And these postbiotic metabolites are acid stable. They're not living organisms like bacteria. So they survive transit through the gut and the small intestine. And when they reach the colon, they rebalance and reestablish the whole ecosystem. But back to SIBO, my studying of the literature and talking with doctors that treat it, maybe 20% of people with SIBO can cure themselves on the FODMAP or the specific carbohydrate diet. Most people are unsuccessful, and they have to use the antibiotic rifaximin, and that clears out the gut. But then they have to really pay attention to rebuilding a healthy gut microbiome. And they have to make some diet and lifestyle changes so they don't get the same condition back again. So they really have to make dietary changes and get into doing my salad buzz to get all this diversity of plant-based foods in their system. And they have to exercise regularly and get physical movement. And so diet and lifestyle changes following the rifaximin protocol are really required so that the SIBO doesn't come back again. But for people taking this fermentation product I've talked about and recommended this evening, Most people with SIBO can tolerate it, but I encourage people with SIBO, start out taking one capsule every other day for a couple of days. If you feel fine and don't feel any worsening of your symptoms, start taking one capsule daily. Do that for a couple of days. And if you're okay, then go up to the recommended dose, which is 2 capsules daily.
Okay. What is your opinion on FMT? So is that
FMT.
I'm not sure what that is exactly.
Fecal transplant.
Oh, fecal transplants. Yeah. Fecal microbial transplants are astounding. They've been shown to be probably the fastest way to change really severe intestinal and gut problems. There are now centers that are being certified to take fecal samples from, they do screening of donors to make sure it's a healthy donor, and then they process the fecal samples, and oftentimes, and most of the time, I think they're producing, they dry it and produce capsules. But a lot of people just home brew it. You got a healthy family member, they'll take a fecal sample and put it in a blender and use a food baster or something to insert it in the rectum after a bowel movement, so there's room in there to insert it. I don't promote that, but I know a lot of people do it. But there's a growing industry of certification for fecal microbial transplant companies that are doing that. It's really quite an astounding procedure. I mean, I've seen people go from like 220 pounds down to 115, 120. Their skin and bones they're not keeping any body weight. They can't digest food at all. Fecal microbial transplant, overnight, it changes. So it's really quite dramatic.
Dr. San Juan says there's a similar OMX made by O'Hara in the Philippine market. She wonders if it's the same product. Is that?
I'd have to look at them. I don't know. I'd have to have somebody send it to me.
Your opinion on keystone species of the microbiome? I'm not sure about that.
Yeah, there are keystone species. These are species that in relatively small numbers can have very significant metabolic effects. I think this Enterococcus faecalis TH10 that I talked about is certainly a keystone species. There's another strain that I didn't talk about tonight, different topic, but there's a strain of bacteria called Lactobacillus fermentum ME3. It synthesizes glutathione. It's the most effective way of boosting glutathione that I know of. Because this strain of bacteria produces glutathione, glutathione is a postbiotic metabolite of that particular species of bacteria. So it's, I think that might be classified as a keystone strain. I don't think we know enough about these, what should be classified as a keystone strain, but I'm convinced of the concept that there are some strains of bacteria that are incredibly important and even in lower numbers have significant biological effects.
Okay. What is your opinion on drinking food grade hydrogen peroxide?
I started a business years ago marketing 35% food grade hydrogen peroxide. It's got tremendous benefits. I have to be very careful with it because it's terribly caustic. And it's harder and harder to get food grade hydrogen peroxide, 35% food grade now. I've used it for probably, 40 years, maybe long. Early in my career, I lived in Mexico for 6 years, and I was hospital administrator of an alternative non-toxic cancer hospital. And one of the primary protocols is that we did intravenous low-dose hydrogen peroxide. Every other day, people were in the infusion room getting intravenous hydrogen peroxide. So that's when I learned about hydrogen peroxide. I've used it ever since. In the last year, I've been unable to find a source of 35% food grade, so I now use 15% hydrogen peroxide. I'll put peroxide in a bowl of water and wash all my vegetables and process my vegetables. And Sometimes I'll gargle with it if there's a little mouth sore or something. But just have to be very careful with it because the food grade 35% is extremely caustic.
This is just, I guess, a statement. SIBO caused by dysfunction of the ileocecal valve.
Maybe. I don't, there may be multiple dysfunctions. I don't know. I think that's certainly a contributing problem, but I haven't studied it enough to know what's known about the true causes.
Okay. And then how do you dilute the food grade 35%?
Just with distilled water.
Okay, so like 35 to 15, so it'd be about 4 parts to 6, something like.
Yeah.
Okay. That's all the chat questions. Dr. Nario has some online one or email questions. So are you there, Dr. Nario?
Yes. Dr. Pelton, thank you so much for the presentation. So I'm your airport correspondent. So if you hear announcements here, it's part of my background.
That's all right.
So yeah. And I have to comment on your peroxide use because that's something that I'm doing also in our clinic. So I definitely vouch for that in so many ways, not only for GI symptoms, but systemic infections. That's definitely something that we go for. And also now on my personal end, I was just in the IFM conference recently, and there's a company there that actually is now customizing probiotics in relation to what specific strains you need for your system. I don't know if you heard of this company and what your thoughts on this customized and personalized approach to probiotic supplementation.
I'm not familiar with that, but my general comment is customization of probiotics. Oral probiotics don't work. It's the postbiotic, but most people taking oral probiotics, they're not going to survive transit through the gastric acid. Most people are not consuming a diverse range of plant-based materials that are the food for the probiotic. So I just don't think a customized probiotic is really is going to be very effective. I think.
Yeah, so they can conduct these specific testing on stool samples of every patient, and they would find the diversity of these results for different individuals are basically the basis of them choosing specific probiotic strains. That's why the ranges of pricing are different in relation to what they find. So it's just something new and innovative that is out there. And I have heard your theory before also that sometimes when they reach the gut, they're dead. And it's the immune response that's actually causing the health benefits of such reactions.
I appreciate your comments, and that's good information. What I would like to see done is when the scientists are evaluating these stool samples and finding which strains of probiotics seem to be associated with better health, the next step in the equation is finding out what sources of dietary fibers and polyphenols are the most effective food for that strain of bacteria so that people can consume the right types of food to promote the growth and proliferation of the healthy bacteria that'll produce the postbiotic metabolites.
Right, those are good points. So here's some questions for you in our emails.
I have Dr. Nario,
go ahead.
Can I ask one question?
Go ahead.
Now, can we combine probiotic, prebiotic, and the postbiotic, and what would be the effect?
Yes, you can combine them, but if they're taken orally, the probiotics are probably not going to survive transit through the GI tract, but the prebiotic foods will, and the postbiotic metabolites will.
Okay. Because I was thinking if they could be more potent by doing the 3 of them rather than one.
I guess I missed that.
No, I said I was wondering if combining the 3 of them would be more effective than just doing postbiotic.
Well, a diversity of foods is really important, and a diversity of postbiotic metabolites is really important. A diversity of probiotic bacteria is also really important, but the only way you can get a diversity of probiotic bacteria is by consuming a diverse range of plant-based foods that are supplying the diverse range of fibers and polyphenol. Taking oral probiotics is not going to really affect your gut microbiome. It's not taking oral probiotics doesn't increase or different or cause different types of bacteria to thrive and survive in your gut microbiome.
Do you make a difference between the live ones versus the non-live one? Because I mean, in the probiotic, they have one that you refrigerate and the other one that you don't.
Well, that's a good point. There's a lot of research being done on killed probiotics or heat-treated probiotic. And they're finding out that there's health benefits associated with that, but because it's the compounds that the bacteria create, they're in the bacterial cell. So they're kind of postbiotics, but they haven't been secreted into the system yet. But when you heat treat or lyse the bacteria, it releases all these compounds in the cell, and that can have beneficial effects. And maybe even some of the compounds in the cell surface have some effects. We don't know that yet, but heat-treated probiotic bacteria are being studied for their health benefits. And some people are classifying the compounds in the heat-treated bacteria as postbiotic metabolites, and other people are arguing that that shouldn't be a correct classification.
What about
Thank you.
spore-based probiotics?
Did you say spore-based?
Spore-based probiotics?
Yes, spore-based probiotics can withstand gastric acidity. So they'll pass through the stomach okay and down the small intestine. But when they reach the colon, they're only going to be effective if they can access a diverse range of polyphenols and dietary fibers, because they have to be able to produce postbiotic metabolites to provide any health benefits. It's not the bacteria, it's the compounds they create.
Okay, that's the watchword today, children. Dr. Neri,
Yes. So you mentioned about these probiotics or postbiotics, and I think they were asking, I mean, the specific product that you're pertaining to, should it be without food? Yes, refrigerate. So it's almost like about the intake. Later on, I'm sure you're going to talk about your product, but I think they're asking about this specifically.
Yeah, good question. Well, I mentioned earlier that's a patented design capsule that the product is in. So it can be taken anytime, full stomach, empty stomach, breakfast, lunch, dinner, in the middle of the night, doesn't make any difference. Just get those postbiotic metabolites in on a daily basis to support and maintain a healthy gut microbiome.
all right.
You mentioned a while ago it should be taken at night. In one of your slides, it was mentioned here. Why is that?
No, I don't remember having that on the slide, but I take my probiotics every morning. The Lactobacillus fermentum ME3 that produces glutathione, I take that every evening. So, and there's no research that tells me that that's a better way to do it. I do my ME3 bacteria for glutathione production at night because I just figured that the liver does probably more of its detoxification during the rest phase, during sleep. So that's why I do that.
Okay, what is your opinion on baking soda?
For what? Cleaning your mouth,
Yes,
keeping
for
odors out of your
I guess for
refrigerator? What?
I'm just reading the questions here.
I think some people use it to brush their teeth with. We talked about hydrogen peroxide earlier. I used to use a weak solution of hydrogen peroxide to brush my teeth with.
Okay.
What about rectal probiotics?
Yeah, good point. Rectal probiotics are effective. I mean, they're going to, after you have a bowel movement and create more space, doing probiotics rectally will get into the lower end of the colon and be effective. And some people will actually take some of this product that I've spoken about this evening and insert the capsules rectally. And some women do it vaginally.
There's
So you mentioned about, sorry, Dr. Clearfield, go ahead.
And so there's a follow-up here is, how about your opinion on the Budwig diet? You know what that is?
Which one?
Budwig, that was she
Oh,
was
Joanna Budwig?
yeah, she was a German minister of health in the early 50s.
Yeah, I haven't heard her
Up-
name for 50 years. I
We have
remember
we
her,
actually
but I
have
don't know about
we
her
have
diet.
a participant who lives in Austria, and she's feeding me these questions here. So
Yeah. Yeah, I don't remember her diet.
um, yeah. Okay, go ahead, Dr. Narian.
So you mentioned about the prebiotics wanting food to work when it gets to the colon. Does it mean that you have to have your food intake first, then take your probiotics? Is that what you mean?
Oops, say that again now. I didn't
So
quite
you
hear.
mentioned about when the probiotics get to your colon, they need to have food to consume, right?
Yeah, yeah,
To create the SCFAs.
yeah.
It's asking here that should you now recommend that take food first, then take probiotics? Is that a better approach?
I'm not suggesting people take probiotics. If they take food, they're taking the prebiotics. And when those prebiotics get to the colon, the bacteria will break them down to produce the postbiotic metabolites. Did I miss something in your question there?
No, that was a straight up
Okay.
question right there. So yeah, so there's another one here. So we are creatures of habit and we eat almost the same thing every day. So what is the drawback of this in relation to the gut microbiome?
Well, I've emphasized how important it is to consume a diversity of plant-based foods. And you're right, we're creatures of habit, and many people have meat and potatoes and a serving of peas or a serving of broccoli. That's one vegetable. You need to get 10 or 15 different types of fruits and vegetables every day, and almost nobody's doing that. But I emphasize, I said earlier, and I'll repeat this, I try to get people to understand the importance of consuming a little bit of as many different types of fruits and vegetables a day as possible. So rather than having one big serving of vegetables, and this is the beauty of my salad buzz that I showed you. 16 different types of fiber and polyphenol containing vegetables in your salad.
Yep, it does look good. Here's one. I think clarifying your statement, did you mention about there's something bad about that immune response that occurs when probiotics gets into the gut?
No, what I said is that there's some studies that have been published documenting that when people take high dose probiotics, 50 billion, 100 billion, 200 billion, it is so much of one type, it can cause the immune system to mount an alarm reaction. The analogy I use is immigration. Every year, the United States lets thousands of immigrants in. That's a good thing. We've been doing it for 200 years. But if they open the borders, and I know that's a big political problem right now, but if there were no border patrol and we had 50 or 100 million immigrants come into our country, everything would break down. The police department, the fire department, the hospital system, the educational system, the grocery store system, everything would be overburdened and diversity and balance would be so all messed up. So the immigration analogy is similar to taking these high dose probiotics where you have massive doses of one or several strains coming in. Even if they're good, healthy strains of bacteria, it's so much that it throws things out of balance and it can cause your immune system to mount an alarm reaction.
In relation to the Harvard video that you showed,
Yeah.
I think this question was pertaining to, so doctors always recommend complete your 7 to 10 days of antibiotics. And if you don't, you're going to build antibiotic resistance. But in this situation, you showed a video that actually, if you take it longer, then they create more antibiotic resistance. So what would be the difference in these two cases or of scenarios that we just presented?
I'm not specifically recommending that people should not take antibiotics when they're called for. What I'm trying to point out with that video is how important it is for people to be proactive at creating and maintaining a healthy gut microbiome and do everything they can think of to optimize their health so they don't have to take antibiotics. That was the main point of showing that video.
All right, that's all I got, Dr. Bill.
Okay. Someone asked if the probiotics would be more effective if they were in the gastric stable capsules from Japan that you alluded to.
If there is, repeat that, Bill, if they're in
And
what?
probiotics, would they be more effective if they were in these gastric stable capsules that from Japan that you mentioned for the preparations that you have?
They'll be more stable, but when they get to the colon, they still have to locate dietary fibers and polyphenols. And I showed you studies that saying 80 to 95% of people aren't eating anywhere near the diversity of fibers and polyphenols that are required.
Okay. Is there a best time to take the postbiotic?
No, anytime, day, night, full stomach, empty stomach, just take them regularly.
Okay.
They have the polyphenol supplements. If you were to take that with a probiotic or prebiotic or postbiotic, how would that work?
I missed the first part of your question.
I said they have the polyphenol supplements that they sell in the market, right? So if you were to take any of the probiotic or postbiotic with the polyphenols that are encapsulated, would that work or not?
Well, it's breaking up a little bit, and I'm missing part of your question. Bill, can you repeat that? I might be.
I'm not try it one more time, Dr. Tano.
Yeah, I said, they sell polyphenol supplements
When you
in
say
the market.
supplements?
Polyphenols.
Polyphenol. Okay.
They sell them in the market. So if these polyphenols are such good for the postbiotic, can we just buy them and
okay.
use?
I got it. Yeah.
Yes.
Yeah. Okay. Yes, there are a lot of polyphenols that are being marketed, things like quercetin, but the thing to understand about polyphenols, I mentioned there's over 8,000 of them that have been structurally identified. Many of them are massively huge molecules, and we don't effectively digest and metabolize many of these polyphenols. So many of them travel all the way through your digestive tract unchanged until they reach the colon. And with the dietary fibers, humans do not contain the enzymes that allow us to digest these dietary fibers. So they also pass all the way through your GI tract, but when these polyphenols and dietary fibers reach the colon, they are the food for your probiotic bacteria. And different bacteria have different DNA, which allows them to digest fibers and polyphenols differently and produce different types of postbiotic metabolites.
Okay.
How does the shikimate pathway contribute or may contribute to all of this?
I missed that one.
Yeah, so how can the shikimate pathway that some bacteria use aromatic amino acids to produce several things like neurotransmitters, how can they help us in this whole process?
Well, I apologize. I'm having trouble with the accent there. You talk something about the pathways.
Yeah, but my internet is kind of not well, that's why. I'm talking about the shikimate pathway, that the bacteria use to change the aromatic amino acid to neurotransmitters and everything else. Can that contribute to metabolizing these postbiotics?
Oh, so how do they do that?
Right.
They have these probiotic bacteria have compounds called Kzymes, C-A-Z as in zebra, Y-M-E-S. Kzymes stand for carbohydrate. I forgot the second word. Carbohydrate appropriate enzymes. They're the enzymes that allow the bacteria to metabolize these carbohydrates and break them down into smaller compounds. So different bacteria have different Kzymes, different compounds that their DNA allows them to produce these enzymes that allow them to digest the carbohydrates and turn them into postbiotic metabolites. So it's a whole new area we're starting to learn about, but it's the enzymes that bacteria have based on their DNA that allow them to create the enzymes that allow them to digest and metabolize different carbohydrates and create the postbiotic metabolites.
Thank you.
Uh, so it's that Minnesota accent from Dr. Tano that gets it. Does it help to use simethicone, especially with gas caused by drugs?
Simethicone, help to use simethicone to do what?
For gas caused by drugs.
Oh, for gas. Yeah, simethicone is a good product that helps with gas. There's another product that's, I think, the best product in the world. I don't have any connection to the company. The company's name is RX Vitamins, and the product is named Acid Block, and it contains two components. It contains DGL, deglycerinated licorice, and alginate, sodium alginate. And it comes in large wafers, and you chew it, it mixes with your saliva, and the DGL coats the lining of the esophagus. It turns into kind of a slimy mass, and you swallow it, and it coats the lining of your esophagus, which protects those cells in the lower end of the esophagus from acid if there is some reflux. But the other component, sodium alginate, when it gets into your stomach, it forms millions of tiny little bubbles that float on the surface of the contents of your stomach. And it kind of creates a foamy barrier that prevents things from refluxing. So it's a very, very effective product. Some people that have bad reflux will chew these tablets 6 or 7 or 8 times a day. A lot of people with reflux suffer from it more frequently when they lie down at night at bedtime. So they'll chew one of these tablets before they lie down going to bed. This is available online. There's a bunch of different companies that market it. It's called Acid Block by RX Vitamins. And Bill, I'll send you I've got an image, a power, or actually it's, I think, a Word document that has an image of this product and an image of how it works in the stomach. So I'll just send that to you, and you can post it for the other attendees to have access to.
So just to remind our folks too, deglycerinated licorice is also an aromatase inhibitor, and it will raise testosterone levels, and it will help control estrogen levels. That just as an aside. So you mentioned it. That's just a little bonus. Okay. So okay. Saccharomyces boulardii.
Good product. It's I think it's not a bacteria. I think it's a strain of yeast, I think, but it's got some very effective. It's a lot of people take it and get some benefits from it. And I haven't studied it for a long time, but I know has some pretty good therapeutic benefits when people use it.
Yeah. Okay. And again, Teresa in Vienna, have you had any contact or any information on there's a technique called frequency specific microcurrent that seems to help that with GI symptoms. So it's microcurrent, applied, different microcurrents applied to different areas to sort of
That's something that's totally new to me. It sounds very interesting. So I'd love to receive some information about it.
Okay, I can send that to you. I've actually spoken at their conferences. They draw me in to talk about head injuries. So, but it's a whole thing that I don't personally deal with. But
I'd love to learn about it.
The Ens Medica has an alginate product, I guess.
Enzymatica? Yeah.
Enzymatica, yeah. Okay. That's pretty much all the comments. Anybody else have any other questions? I know you've been on for quite a while, Ross. Do you have nobody else has any other comments? Could you take a couple of questions about
I,
rapamycin?
I have a quick question, Dr. Bill.
Go ahead.
Now, how does the vaginal canal, the short passage of the baby through the vaginal canal, improve so much the gut bacteria versus doing C-section?
I missed part of that. So try one more time and let me listen closely.
I'm saying, how does the passage of a baby through the vaginal canal
Uh-huh.
actually improves the bacteria, gut bacteria, so much through life?
I think you're asking about a vaginal birth and how does the baby get exposed to the bacteria and how does that help form a healthy microbiome? Is that the question?
That's correct. How does that
Okay.
help them?
So when
Because it's
baby,
a short passage, it's a short time. It's not like hours after hours. It's a really very brief passage, and
Yeah.
yet it conveys so much potency.
Yeah. So when the infant travels down the vaginal canal, it gets covered in mucus. And most of us have seen a baby born and it's got this mucus all over their body and the nurses start wiping them off. But some of that mucus gets in the infant's mouth and they swallow it and it gets into the digestive tract. And that's their first exposure to healthy human bacteria from the mother's vaginal microbiome. And that will start to grow and proliferate and is critical for the formation of a healthy microbiome in the infant.
Okay.
Got it.
Okay. Okay, great. So do you have like just a couple minutes for?
Yeah, Bill, you got a couple questions about
Well,
rapamycin?
is there a protocol? Is there a laboratory? I know the sirolimus, lab test, but is there a protocol, sort of a general overall protocol that
Or taking rapamycin?
well, yeah, if somebody comes to you for a longevity evaluation.
Yeah, well, again, this is a new frontier. There is not enough research that has been done, but I'd say probably, I'm guessing that 85 to 90% of people who are taking rapamycin are taking 5 or 6 milligrams once a week. That's the study that Joan Manick did with elderly people, showing that 5 milligrams once a week got about a 20% boost to the effectiveness or the function of their immune system. So that's what most people are doing. If you go on the website rapamycin.news, there's a lot of people that are putting in comments about trying some different protocols. For about the last 6 months, I experimented taking 12 milligrams once every 2 weeks, did blood tests and everything. Didn't notice much difference. My theory behind that is that a little higher dose initially is going to get a little higher activation of autophagy. But again, we really haven't done the studies on this to really fine tune what's the best dose and what's the best frequency of dosing. But I really like to emphasize to people how important autophagy is. This is the big missing piece that people need to understand. For hundreds of thousands of years, people evolved eating probably one meal a day and munching a little bit. But if you eat one meal a day, you spend about 4 hours digesting your food and 20 hours a day with no food intake. So for most of human evolution, people spent most of their time every single day in autophagy. Now people are eating breakfast, lunch, dinner, in between meal snacks, dessert, nighttime cocktails. It's totally reversed. These days, people spend almost all their time with mTOR activation and not adequate time in autophagy. And autophagy is the process of breaking down your body's old dysfunctional parts and rebuilding with new parts. And the analogy I use is a car with a flat tire. You can't put the new tire on until you take the old tire off. But throughout your body, you've got enzymes and proteins and lipids in your cellular membranes that are damaged, but they're taking up space and they're not functioning. Autophagy allows you to start to recycle those and build healthy new cellular membranes and proteins and enzymes and so forth. So that's why autophagy is so critically important, and people need to spend more attention about spending more time without nutrients coming in. And a lot of people aren't going to discipline themselves to do intermittent fasting, and rapamycin does it for you. Goes into the cell, binds to mTOR, partially inhibits it, so you can activate autophagy.
Could you explain again the rationale or the fact that to build muscle you need to activate mTOR, but somehow the rapamycin, which inhibits mTOR, seems to help with muscle growth.
You bet. So when rapamycin first became available, a lot of bodybuilders and high-end athletes didn't want anything to do with it because it's inhibiting mTOR, and mTOR is the signal for growth. The analogy I use is a Formula One race car, really, really fast. But you can't drive that car fast forever. You'll burn it up. Just like humans, you've got people that are running a 100-yard dash or a 440-yard dash. They can run fast for short periods of time, but you have to have down periods of rest and relaxation to rebuild. And so this is why autophagy is so important. You want to have autophagy activated so you can break down old dysfunctional muscles and cellular components and membranes and so forth, so that then when mTOR gets activated, you're replacing the old damaged things that have just been broken down and recycled.
Okay, that's why
without that you never get autophagy, and people are missing out on the rebuilding of their body, and they're accelerating the aging process.
Okay. And 6 milligram weekly dose. How long before someone would notice a difference?
That's a great question. And my way of answering that is, when you take a dose of vitamin D today, how much different do you feel tomorrow? Or vitamin C or whatever. You don't feel any different. You take it because you understand the science behind it. And the most noticeable effect that some people have experienced is weight loss, predominantly visceral abdominal fat. That's been reported in animal studies. But on a day-to-day basis, people aren't going to notice much of anything. But when you start taking rapamycin every single day, day by day, every single cell in your body starts to work better. But it's a slow, gradual process. It's not about quick benefits. It's about the long-term process of slowing down the process of biological aging and optimizing your health.
So what, let's say you've been doing your 12 milligram every other week for, you said 6 months?
I did that for about 6 months. I'm back now to the weekly regime again, just going back and forth. And one thing is frustrating for me, I used to have a LabCorp phlebotomy station close to my house, with just like 10 minutes away. They've gone out of business. So now it's not easy for me to get my blood draws and track things as it was before. Before that, I was getting blood draws with every dose and 20 hours after a dose to see how fast it's absorbed and metabolized. But I can't do that anymore.
Okay. Well, I think I'm good.
Okay. I know you're good.
Anybody else have
appreciate
any?
everybody's interest this evening. And Bill,
Thank
if you'll
you.
get me the attendance list, we'll get some samples of
certainly.
products sent out to all the people that live in the United States.
I will get it to you as soon as I can get it to you.
Yeah, no hurry, no hurry.
Anybody else have
Appreciate
any questions, comments? Anybody?
Can we get contact information?
My website is naturalpharmacist.net, and my email is rosspelton70 gmail.com. And I have to just, please don't flood me with emails because I get a massive number of emails and I try to answer them all. But if there's 70 of you all sending me 2 or 3 emails a week, it's going to put me on overload.
Sorry, what was it? Ross Pelton, what?
I have it in the
70,
chat.
70,
Oh.
Ross Pelton 70
Oh, okay, thanks.
@gmail.com.
I have it in the chat. So, and
Got it. Thank you.
All right, everybody. Thanks very much. Appreciate your interest.
thank you so much. Again, being as competent and interesting as you are, we're gonna have to do the disservice of asking you to return. So,
Okay. I look forward to it, Bill.
okay.
Thanks,
All right.
Mike.
Anybody have any
Bye, everybody.
comments, questions? Thank you so much, Ross. It was really great.
Bye now.
Okay. Bye-bye.